Okay. Well, thank you so much for joining me. And thank you, Fran, for the invitation. I'm really delighted. This will be my first remote seminar, research seminar that I've given. It's also the first time I'm going to talk about a lot of the stuff I've talked about with singing mice. I'm trying out a few new things. I hope that you will excuse me if a few pieces are rough. I always tell my students not to start off with an apology, and that's exactly what I've done. But no, I say that actually because I would look forward to any feedback that you have, and I don't mind being interrupted either. So if you want to raise your hand, I'll try and keep a lookout on the on the participants, um, and I'm happy to take questions as I go. I'm also going to be talking about a new kind of conceptual framing of some of our work that I think has some rough edges too. Okay, so the title of my talk is Making Sound Decisions, Singing Mice as Integrative Models for Adaptive Display. And so I should tell you maybe a little bit about myself to begin. I would call myself an evolutionary neurobiologist. I am very serious about animal behavior, but I'm also very interested in neuroscience and molecular mechanisms of behavior. And I'm particularly interested in kind of a really interdisciplinary, integrated view on how behaviors evolve. And so for me, a really satisfying explanation uses evolutionary principles to organize our ideas, but then gets down to more detailed mechanisms underlying those behaviors and seeing how those mechanisms themselves evolve. And for much of our work, I usually talk about our work with prairie voles who form pair bonds, but today I'm going to talk about singing mice. And singing mice are a model system that we've been developing as a lab since I first started my lab in 2002. But when we first began, we didn't really know anything about them, including basic husbandry. And so we spent a lot of time learning their natural history, kind of describing aspects of their brains. And I feel like we're starting to get some real traction and have a kind of critical mass of work. And so I'm happy to finally be assembling it for you today. So when I think of behaviors, I think of them as instantiations of adaptive decision-making processes. And I think of the brain as being tasked with selecting actions that are going to likely to provide the best fitness outcome. And the best fitness outcome is something that minimizes risks while maximizing rewards. And so risks and rewards, for example, in the context of a display, like this vocalization by a Tungra frog, Phycelamus pustulosus shown here. This is a frog that Mike Ryan in my department is famous for. Mike also happens to have been my dissertation advisor, and I worked on these guys myself once upon a time. And so male Tungra frogs, like lots of frogs, call. And so when they vocalize, the hope is they're going to attract a female and that they'll be able to set up the that they'll be able to grasp a female and then build a foam nest to house their eggs together. But there's a very real chance that when they call, they'll be eaten by Trachops, a frog-eating bat. And so males need to assess the environment as it's changing. They have lots of sort of rules they seem to use for making those assessments and maximizing their fitness. And of course, that's true across the board in all kinds of animal behaviors. And I think displays are really eye-catching or really dramatic examples of behaviors because they're meant to be noticed. And they face these kinds of constraints really conspicuously, and a lot of animal behavior is concerned with understanding aspects of display. Well, when you think about whether or not to display or whether or not to do any behavior, I think a really good framework for thinking about it is kind of Bayesian decision-making specifically. And what I mean by that is you are probably aware of Bayesian statistics or Bayes' theorem. And the idea is that you're trying to estimate the state of the world in some way based on prior information and based on current information. And Bayesian decision-making takes that framework for thinking about it, certainty and uncertainty, or degrees of uncertainty, and adds to it a kind of what was historically a signal detection theory framework that thinks about costs and benefits and how you balance those with uncertainty. And one important thing I think about a Bayesian framework is it really kind of emphasizes that information that you use to make good decisions accumulates across time. And I think it's a really interesting way to think about the structure of behavior, but also to think about the structure of the nervous system. So I'm going to kind of like try out this new framework, a new phenomenology, just drawing from kind of the way a psychologist might talk about these kinds of things. So I'm going to describe traits as aspects of the phenotype that are stable across a lifetime, and for us, that's going to include behavioral proclivities. So those could be either genetic in origin or developmental in origin. And if they're developmental in origin or genetic in origin, it might include things like animal personality, for example, things that are personality usually is common across lots of contexts, but anything that's a stable characteristic of an individual. Well, that may set up a kind of baseline of behavioral performance for a given individual, but then that gets modified by the state that you're in. So the states are things that I define as varying within a lifetime on time scales from minutes to years. And you can see there's a lot of sloppiness here. States often depend on interoceptive cues. And states kind of rely on prior information about what the relationship, maybe not prior information about what the state of the world is right now, but prior information about what the best thing to do is given the state of the world. So I think of traits as being kind of priors, Bayesian priors that you inherit or that get developed, get tuned during development in a way that we expect to be adaptive. And then states as being kind of adaptive responses to changes in the state of the world, but changes that you could predict based on your evolutionary history in some way. And so changes in, for example, if you're in a fearful state because you've heard something that sounds like a predator, then there are things you would know not to do, right? You don't need to learn not to display or anything like that. Those are things I would consider states, and they often depend on interoceptive cues, things that you might do naturally in response to hormone release, for example. And like responses to interoceptive cues, they might have a range of time spans, but things that vary, say in hormonal language, like activational effects, for example. And then the third thing I'm gonna suggest is that there's another aspect of adaptive decision making, which I would call not states or traits, but situations or situational decision making. These are things that are really flexible and dynamic, and they occur often on a time scale of say milliseconds to hours. They depend on external cues, and they're not things that you could have predicted. The relationship between the world and the best behavior you should exhibit is idiosyncratic and depends on experience. And so conditioning mechanisms are ways of updating our behavior in response to our baseline behavior from our traits, and then our state-based behavior. Then we get further refined through learning mechanisms that then allow us to fine-tune things in a way we just couldn't have put into the genome, right? There are too many unknown unknowns in the world not to have these kinds of mechanisms. So that's that's the general kind of conceptual framework I'm going to forward. And I'm going to suggest that this conceptual framework has some consequences for how we think about the brain and its role in adaptive behavior. And so these are a list of brain regions, which I don't expect you to know. I'll come back to a couple of them a bit. There's not going to be a lot of brain work in the talk today, some. But these are brain regions that are known, were described by Sarah Newman in 1999 as a social behavior network. And she noticed that if you looked across literature for lots of different kinds of social behavior, parental care, aggression, sexual behavior, now pair bonding, I would add to that, but there are a handful of brain regions that just keep showing up again and again. The lateral septum, the medial amygdala, the bed nucleus of the stria terminalis, these are all areas that might be collectively called the extended amygdala, that is the amygdala plus its related structures. Then there are a bunch of hypothalamic areas, the anterior hypothalamus, the ventral medial hypothalamus, the preoptic area, the paraventricular nucleus, that kept turning up in lots of behavioral studies, social studies. And then the periaqueductal gray, which is a midbrain area that seems to gate a lot of emotional responses or things that are considered instinctive responses. And so these brain regions kept turning up time and again, and I'd suggest that they kind of map on loosely to some of these ideas. And what I would say is that the function of hypothalamic structures is for the most part to regulate our state. That is, they send descending projections to brainstem. They can influence our arousal. They secrete hormones that have transient but significant effects on the behavioral decisions we make. And so we use that to integrate information about daylight, about our reproductive state, about our fearfulness. Those things are all happening at some level in the hypothalamus. But the hypothalamus gets lots of inputs from the extended amygdala, and the extended amygdala is extensively implicated in learning. Fear learning, social memory, social recognition, all kinds of things like that. And so I'm going to suggest that this kind of extended amygdala part of this network is the part that's connecting this situational information that you can't really encode in the genome or in the brain, from scratch. It has to be learned. It's taking those contingencies and giving them access to our states as well as to our other behaviors more directly. And then on top of that, we have sensation and memory feeding into these structures and providing the cues we can use to make those predictions. But then those things also can have, through other pathways, through cortical pathways in mammals, descending projections on motor function that bypass these kind of state-based mechanisms and have more direct control over situational responses. So that's kind of the general conceptual framework from behavioral ecology standpoint, and then again from a more systems neuroscience standpoint. We'll come back to these again at the end. Well, when a behavioral ecologist thinks about regulating display, we often think about the trade-offs animals face between costs and benefits. And for me, this Johnstone paper from 97 or 98, I forget, that's, my slide says 97, I'm gonna trust it. I think is a really just great summary of how we think about how elaborate to make a display. So if we think of signal intensity as something the signaler, usually a male, has control over, and then there are costs and benefits to fitness based on having that signal intensity. Well, the more intense the signal, the greater the benefits are, because you're communicating something for a reason, right? That you're trying to adjust the behavior of somebody who's listening to your display. But those benefits don't accrue linearly, they taper. The costs, however, in what Johnstone calls a cost-scaled system, the costs may shift in their slope. So for some animals, the cost may be low, or some context, the cost may be low, and for others, high. And so, and then depending on which it is, the optimum difference between those two shifts. And so in this low-cost case, in this, I think you all are seeing what I'm seeing. I'm just realizing this L is beneath something that's kind of steeply scaled. I'm not sure why that is, but never mind for us, because the point is that differences in the slope of the cost function means that we're going to have differences in the intensity. So if the costs are relatively low for a given intensity, then we're going to make a more intense signal. If the costs are relatively high for a given intensity, then we're going to make a lower intense signal. And that just makes sense. And that includes things like predation risk. So if you've just seen a predator, you might shift your perceived costs. You might also do it based on state, like based on your developmental environment. You might alter your stress reactivity throughout your lifetime. I would also include, say, body condition. How much can I afford to invest in this display if I'm making something expensive, but also how much could I afford to expend on a display if it's going to cost me to produce it, or if it's going to entail costs of injury, for example. We can talk about benefit scaled systems, as Johnstone did. And so these are similar, except the cost structure is the same, but the benefits shift by context. And in this case, then we have kind of a similar calculus that might govern our signal elaboration. And I see now why the L and H is for. It refers to whether the signal is low intensity or high intensity, not the cost. Anyway, so this could include things like reproductive status, whether you're male or female, whether you're immature or mature, whether you're reproductively quiescent or not, variation in social context. All these things are things you might expect could have an effect. And in fact, that seems to be the case for lots of animals. And that seems to be the case also for many species that display vocalization. We're going to talk about vocalization as our focus. I began with the timber frog, but that's the last you're going to hear about those lovely little animals. But vocalization in general is really interesting because it's a very widely distributed behavior. It has obvious trade-offs in the context of predation, reproduction, social interaction, and it's been well studied both mechanistically and ultimately. We also know something about the distribution of vocalization, know that among vertebrates, that's very ancient origin. So evolved only a few times. And we know a good bit about some of the neural mechanisms of vocalization that, some pattern generators seem to be common for lots of displays across taxa, etc. So we're not just going to talk about vocalization in general. We're going to talk really specifically about singing mice. And who are singing mice? Well, singing mice are chrysetid rodents, but they're New World, and they're relatively closely related to deer mice, not particularly closely. And they're called singing mice because I'm just going to check and make sure I got a text. I just wanted to make sure it wasn't somebody texting me to tell me my internet was out and I didn't realize. Okay, so singing mice are fascinating little beasts that live in the cloud forests of Central America. And this is a singing mouse here, Scotinomys tiguina. This is another singing mouse, not the other species, Xeromplinus. And then this is a sister taxon, Bayamese taylori, and this is called the pygmy mouse. And these occur in the Southwest US, including here in Texas. And all of these make these kind of trill-like vocalizations. And so what we have here on the y-axis is the frequency of the song, of the vocalization is going from 0 kilohertz to 100 kilohertz. So these are really high frequency ranges. And what we see is that the two species of Bayamese, which are the sister genus, make this kind of frequency modulated trill with, this is about 2 seconds long. And it has, compared to these guys, a relatively narrow bandwidth. There's a relatively narrow range of frequencies. It's entirely ultrasonic, like other muroid rodents. And the repetition rate of the trill is relatively slow. And it's not that slow. It's, 20 times in 2 seconds, something like that. But if you were to compare that to the two Scotinomys, their repetition rates are much faster. In fact, these songs have been squished onto these representations that aren't really great for them because they're repeating these notes so quickly and going across such a bandwidth that you might think that these are not frequency modulated. But I'll show you a detail here in a moment, and they are. So Xeromplinus makes also a song a couple seconds long, but tiguina seems to have this derived feature of a particularly long song. So shown here is a little short song followed by a full song, not a full song, a few seconds of song. Their songs are generally around 6 or 7 seconds long. The longest one we've recorded is 16 seconds. And the length of the song varies somewhat from population to population. There seems to be a genetic contribution to it. We've done some phylogeography of the singing mice. This is from the dissertation of my student Jorge Pino, who's at the University of David in Panama now. And this is just sort of showing major lineages of the mice and their distribution across Central America here. And so here's Costa Rica down here, and this is Panama. And then we've worked a bit here and then also at some of these sites here. For the most part. So most of our animals that we work with these days come from these Costa Rican populations that are actually sympatric between the two species, as we'll mention briefly later. I'm going to show you a couple of videos, and to do that, I'm going to need to share my screen in a different way. I'll share this. There we go. Okay. Okay. You guys see my window? think you can. So they get up on their hind legs and they make these little songs, and that's one example. Um, so they're very high pitched, and the recording isn't fantastic because they're higher pitched than regular microphones do well at. But if we slow it down just a little bit, just threefold, and so this is a video that was captured at 100 frames per second and played back at 30 frames per second, and the sound adjusted correspondingly. And you'll see the difference, I believe. You can hear it too. It's majestic, isn't it? Not everyone would call it that, but hold on here. I'm going to play it again. This time I'm going to talk through it. I want you to notice a couple things. So with each note, he's taking a little breath. You can see it as the notes get slower, but you can also see it down here. And then with each note, he's also opening and closing his mouth. And then what you can't see is in here in his larynx, he's contracting his cricothyroid muscle and probably some other muscles that are adjusting, that are contributing to frequency modulation of the note. And so, so we have this very interesting behavior, and the song is now much longer, it's louder, it's lower in frequency, and those are all features that increases active space. And in fact, we know from a variety of things that different folks in my lab have done, Polly Campbell, Brett Pash, Aaron Gilio, that they make these songs if they hear another male, or a really good way to evoke the songs is to present them with a male or female and then take that animal away. They sing much more if you give them the social context of knowing that there's an audience, but the audience not being near. If the audience is near, especially if it's a female, they'll increase their songs for females, but they really like to make these more quiet, short songs and things that resemble ultrasonic vocalizations of other species when they're face to face. So these mice are diurnal, which is kind of a fascinating thing. In fact, our hypothesis for the evolution of song is that the mice, in transitioning from being nocturnal to diurnal, were freed from most mammalian predators, and then dealt mostly with avian predators. But the avian ear doesn't have the same design as the mammalian ear, so the avian ear can't really hear these frequencies, even though they're much lower than normal mice, allowing them to communicate at great distances, 20 meters or so, it seems, in the field. So here's a better depiction of what the song looks like. And so this is frequency, and so this is tens of kilohertz. So it's still 100 kilohertz at the top and 0 at the bottom. And so this is a sonogram here. And so 0 to 7 seconds. This is a more normal length song. And then if you zoom in on one of these windows, each of these windows is just 20 milliseconds. And so in 20 milliseconds here at the beginning of the song, you can see 4 full notes. I'm sorry, it's 200 milliseconds. It's 20 milliseconds when I'm zoomed in here. Is that right? Yeah, that's right. And so, so we get these repeated notes, and so you can see how frequency modulated they are, and then you can see that as the song progresses, the notes get a little bit higher in their bandwidth. The max frequency gets a little higher, and then as they go on, the notes get kind of a little more elongate as well, as does the interval between the notes. And so we can pull out just one of those notes, and so this is 20 milliseconds. And look at a sonogram or spectrogram just of the note here, and we could take the maximum energy for each of those find the frequency that has the maximum energy in each of these little bits of time. And we can plot that, and we can fit a little curve to it that very nicely describes the modulation of a note. We could take that same note and look at the amplitude envelope and measure the timing of this first peak and the size of that first peak, and then measure the overall peak and the size of and the timing of that. And when we do those things, we get this the slope of the note, and so the note shape is being governed by the starting slope here, this B term, and the curvature of the note, this A term. And so once we've described the amplitude envelope with a few measures and the frequency modulation with a few measures, we could describe a note in enough detail to synthesize it from scratch. Well, it just so happens that all the features of the note then change systematically with the song. And so I'm plotting here note duration as a function of note number, where 0 is the beginning of the song. And you see it changes really reliably over the course of the song. And it's not just true of note duration, it's true of all the variables we might measure. And so here are a bunch of those I've just talked about. And so the great thing about this is, even though it's kind of an elaborate vocalization for a mouse, it has a real clear pattern to it, and we can describe it really exhaustively. The downside is, if you describe it exhaustively, then you've got lots of variables then that you have to kind of make sense of. And we'll see that, but that's not too bad. So one thing you can do to reduce the number of dimensions of all these things is to do a principal components analysis. And we've done this with multiple data sets now. We've done this with data sets that are across lots of populations. We've done this with data sets from wild-caught animals within a population. And then we've done it in the lab with animals that have been exposed to different social contexts. And every time we look, we see a pretty common pattern. So this is from Burkhardt et al. 2018. The first component of the variation is something that we're calling song effort. And so it loads highly on the length of the song, the number of notes in the song, and song length in seconds, the total number of songs or the song rate, as well as some other features. And to us, this looks like how motivated the animals are to sing, or how much they think they should be singing. The other dimension that loads highly as we look across songs is frequency modulation. And that frequency modulation is governed by the very starting frequency, or the maximum frequency of the song. And then the note slope and curvature, and how that changes over the course of song. And interestingly, that frequency modulation is much more distinctive of individuals, contains a lot of potential information about individual identity, but we haven't found it to be related to any functional features, in the sense that it doesn't seem to be changing with body condition or reproductive state or any of the other things I'm going to talk about. And we think that this frequency modulation aspect, these features here in particular, are markers of individual identity, but we don't have any data other than their distinctiveness to support that. But certainly we're looking forward to investigating that. Here's an example of a high effort song. You see it's relatively long, and these are scaled. They have the same peak amplitude in the recordings, but you see it has a long, slow crescendo, and this one lasts about 8 seconds, whereas the low effort song here is one that lasts much shorter, a little over 5 seconds, and then has changes in the course of the amplitude modulation as well. So that seems to be a major dimension of variation in how animals sing. Well, what else? What are the kind of factors that might influence something like song effort? Well, we've done a lot of different experiments over the years that we can kind of summarize here briefly. One of the first things we did was to pass a model hawk over singing mice. And the reason was, when we first started to work on these guys, folks kept asking us, well, how do you know it's not an alarm call? I mean, kind of knew it wasn't, just because of the nature of it. You know, you don't make an alarm call that's 16 seconds long. Anyway, but we thought it was important to test. And so we showed that if you put mice in an open field and pass a model hawk over them, they will suppress their rate of song. and freeze in response to this increase in predation risk, something we might call a state change. But we also have other kinds of trait level changes in predation risk, like animals that sing spontaneously when we catch them in from the wild are less stress reactive. We find that they have, I'm sorry, yeah, they are less stress reactive. They're more bold in open field tests. They have they have more modest court responses to restraint stress. And those same less stress reactive animals sing more when we bring them in. It's clear the song is involved in mate attraction. If we play back songs, females approach song. Females show preferences for certain songs over others. Brett Pash, my graduate student, showed that. It's also true that females evoke song. In fact, one of the best ways to get a male to sing is to present him with a female and then remove the female. Then he'll sing like crazy. We also see songs being important in male-male competition. So female removal or male removal also evokes song. Males sing in response to the playback of other male songs. If we, my student Andreas George did some nice stuff where he manipulated the aggressive experience of animals and more animals that have experience winning fights. Again, I would call that a state change. Then increase their rates of song. We see resident animals show positive phonotaxis to intruder song, but in certain interesting ecological circumstances, which I'll mention in a moment, we get negative phonotaxis. That is, they move away from the speaker if we play the songs of dominant males. Singing behavior is influenced by sex and reproductive status. Singing emerges at puberty and is sexually dimorphic, with males singing much more than females. Body condition seems to influence song, as we'll see at some length. And there does seem to be some genetic variation. We know that from sort of population level studies or the phylogeographic studies where we measured songs in lots of populations and showed that within a species, the differences in song between populations were associated with the genetic distances in those species between those populations. And we have some other really interesting data I'll describe at the very end of the talk, just in broad strokes. Okay, so what I'm going to do now is I've given you my conceptual framework that I'm working in, and I very breathlessly ran you through a bunch of details about these very cool animals. What I'm going to do now is I'm going to talk about a couple studies that are interesting that I think are kinds of situational things, that short-term changes, changes with a lifetime of behaviors. And then I'm going to talk at some length about kind of state-based things, specifically reproductive status and energy balance. And then I'm going to talk really briefly about trait level things and heritability that I'm alluding to here before wrapping up. Does anyone have any questions about the mice before I move on and tell you a little bit about a couple fun studies? No? Okay. All right. So then, let's move on. So there are a variety of social situations that influence vocal display. We know we've done lots of things where we manipulate the environment, or we manipulate the social context, like presenting with a male or female, or presenting them and removing them, or removing a different potential audience, and those all influence display. But I'm going to tell you now about a couple of really interesting studies. The first one is not led by my lab. It's our first kind of collaboration that we've done seriously with another lab. And this is Michael Long's lab at New York University. And we've known for a while now that males sing to one another, counter sing to one another. And so if you bring them catch them in the field and you bring them into the lab, that they'll start to sort of sing back and forth in a way. And Michael's group took advantage of this, and they did kind of an interesting elaborate study. And what they did is they brought a mouse into an isolated room and kept him in a chamber and recorded him. And they kept him there for about a week. And then after a week, they'd bring in a new mouse. And so we call that first mouse the resident. And then we call that the new mouse a recruit. And if you're in the field, what you'll find is that they have stable home ranges. They're not very territorial, but they have stable home ranges. And then, occasionally new young adults kind of move into the area. And whenever the mice are young adults, like right after puberty, they kind of overproduce song. They sing like crazy. So you bring this recruit into the room and he sings a lot. And then the resident male will sing back. And in fact, the resident male will interrupt him. And so that's what's being shown here. So the resident is being shown in black and the recruit in red. And the recruit's kind of the focal male in the study. And so the resident starts to sing, the recruit patiently waits till he's done, and then moments, literally about a half second after he finishes, the male starts to sing. Now the recruit sings, and then the resident male says, oh no, and then he starts to sing, and he shuts up. And then he waits until he's done, and then he starts singing again. And this is kind of a general pattern that this science paper of Michael's goes through in some detail. It's a fascinating behavior pattern, but what's really cool about this project is that we were able to localize these effects to a particular brain region. And so through some variety of methods, kind of identifying potential brain regions, Daniel Akobe and Arka Banerjee, who are the joint first authors on this study, systematically probed motor cortex. And there's a region kind of anterior that they discovered that when they stimulated it, they could detect effects on musculature that was related to song. And then they also found that if they stimulated it in the middle of a song, that the corresponding musculature would seize up from the stimulation, and that would interrupt the song. And so they're able then to localize the spot in the cortex. And it was an interesting spot. We called it orofacial motor cortex. It was an interesting spot because it was an area that's thought to it's in a region of cortex that's thought to be involved in kind of sensory motor transformations, which is a kind of fancier technical way of saying something that kind of obscures how interesting it is. How is it that an animal, including a human, takes this really complex situation it finds itself in that it's probably never seen exactly before, and then uses that to update its actions? So very much the kind of thing I would think of as being situational. And so then what they found is this area was in fact causally related to this ability, because if they put a GABAergic agonist, so this is something that will suppress neural function, if they put a GABAergic agonist into this site, that they get a severe depletion of the responses of the resident male. So these are each of the dashes are a playback from a speaker of a resident male, and then this is the recruit response here. And you see lots of responses here, and the responses aren't just in direct response to vocalization, but tend to kind of increase the vocalizations throughout the duration of the playback. This is a 1 hour window here. And then these are the same 6 males, but after the GABAergic blockade of this brain region. So in this case, we're able to localize to a specific brain region the dynamic modification of this behavior. An older study that was done in my lab, it's this really beautiful work by a thesis by Brett Pash, who's now at Northern Arizona University. And he's just a really spectacular behaviorist, mammologist, and overall scientist. And he did this really amazing dissertation. And the highlight was this paper, this enormous paper in American Naturalist. And so I mentioned there are 2 species of singing mice. They live at different altitudes. And Scotinomys xeromplenus, shown here at the X, lives at very high altitudes. It has a large body size. They're both kind of small. The Tiguina is about 12 grams, and xeromplenus is 15 grams or something. They're both small mice, but the xeromplenus are noticeably bigger. They have a larger body size, slower development, and they sing much less than Tiguina. Everything about them, and they're also less dense in the environment, everything about them kind of suggests that they're resource limited. And associated with that, they're also quite a bit more aggressive. Well, as you, in Costa Rica and Western Panama, as you go up the mountain, there's a point, if you set traps alongside, as you go up the mountain, there's a point at which one species stops occurring and the other starts. And what Brett found was, even though the exact elevation of that displacement area varied from mountain to mountain, it was almost always at exactly the same temperature. And he did some really beautiful reciprocal removal experiments. He took out the Xerum plenus and found that if the Xerum plenus were removed, the Tiguina would move into that space. But if he took out the Tiguina, the Xerum plenus would not move into the vacant space. The Xerum plenus were basically going as far down the mountain as their thermal tolerance would allow, as far down the mountain as they cared to. And the Tiguina, on the other hand, were having their ranges limited by the behavior of the Xerum plenus. And that seems to make sense because the Xerum plenus, I said, is really aggressive and larger. And so when Brett staged interactions in the lab, he staged sort of 12 aggression trials between these two species, and the Xerum plenus always won. So he then played back the songs of Xerum plenus and Tiguina in the field, and then again in the lab. And so what we're showing here are in the field, he just recorded how many songs they sang in just a few minutes after playing back a song of either Xerum plenus or Tiguina. And so this is whether or not that species sang to the conspecific song, and they both are likely to sing at roughly equal rates. Whether or not they would sing to the heterospecific song, or whether or not they sang to a noise control stimulus. None of them sang to the noise control. They sang to the conspecific at roughly equal rates, but you see their dominance status is reflected in the likelihood that they'll sing back to the heterospecific song. The Tiguina sing back much less. And if we bring them into the lab and look at these Tiguina and repeat the experiment in a more controlled setting so that we can get more data, we see that not only do they not sing back to the heterospecific, they actually sing back significantly less to the heterospecific than to noise. So they actually suppress their song. Now, these are mice that are captured syntopically. They're at the same site. And Brett was very clever about this because he found a site on the mountain where the forest came down to a particular point, and then it was grassy after that. And basically in the grassland, it was too hot for the Xerum plenus, but the forest was still good. And that gave him a very clean displacement zone, and then he tracked right along that margin and was able to get a mice that seemed to have had experience with one another. When he went to another place where the mice were in allopatry, he found that the allopatric Tiguina did not respond this way to the Xerum plenus song. In fact, they respond to it pretty well. That they treat it like a conspecific, and we see the same thing if we look at their brain. They treat it like a not very good conspecific, like they might sing about 10 songs on average, if I remember correctly, in allopatry. So they don't suppress their song. They don't show negative phototaxis to the heterospecific, which they do in sympatry. Okay, so we looked at the brains of these as well, Brett did, and he found, looking at using immediate early genes to identify what brain regions were active or not, he was only able to look at the auditory system. But what he found was that in both the medial geniculate and the auditory cortex, that both allopatric and sympatric populations responded nicely to the conspecific song in their auditory system, as you would guess, because it's a biologically salient signal. But they differed in how they responded to the heterospecific. And now classic kind of neuroethology would suggest that they should respond to the heterospecific by filtering it out, right, if they're in sympatry, because they don't want to confuse it with conspecific. But in fact, we found the opposite, that in sympatry, they're much more likely to respond to the heterospecific than they were if they were in allopatry. And I think the reason is that in sympatry, it's a really biologically salient signal, and the mice can tell it apart from a conspecific, whereas in allopatry, they just treat it as a conspecific, but not a great one. We don't know that this is the case, but it's our hypothesis, our speculation, that these population differences are due to experience. And so what we'd like to do is go back to the site and sample animals that are in the same sympatric population, but sample animals that are in syntopy with the dominant species, and other animals from that population that are allotopic but still sympatric, and see if we can really attribute the differences in behavior and brain activity to differences in experience, which would make it this kind of idiosyncratic thing that the animals are learning as a function of their ecology. Okay, so I'm going to now shift into talking about some things related to reproductive status and energy balance. They're going to be kind of the heart of the seminar today. And this also involves work from particular grad students. I'll begin by talking about sex and reproductive state. So we've known for a long time that the rate of song is sexually dimorphic. So these are, from data we got from lots of populations where we looked at females and males, and just we have like a standard protocol where between certain hours in the morning, we record them for 2 hours, and then we just record all the songs they sing spontaneously and the number of songs total. So if we do that, we see that, in the absence of playback, about half the males will sing spontaneously, but more like 20% or fewer of the females will sing spontaneously. When we look at the structure of songs, we also find that males sing more loudly, and that the length of the songs themselves are louder. However, if we look at other features of the song, like this frequency modulation I mentioned, we don't really see anything else that's different by sex. In fact, I don't think the mice can tell the sex of who's singing from the song itself. So, where does this sex difference come from? Is it androgens? Is it testosterone or one of its derivatives? And if so, how is it acting? Where in the brain is it acting? So that's what we're going to look at now. My student Brett Pash, who I mentioned a lot because he did a lot of great work, showed that that castration of the males and then androgen replacement, that the castration got rid of these sexually dimorphic features of song, so that the castrated males sang less, more softly, and shorter songs, and then those effects were reversed by treating with DHT. DHT is dihydrotestosterone. It's interesting because not only is it an androgen, but it's an androgen that it can't be converted to another form. So if dihydrotestosterone has an effect on the behavior, then you know that that effect is being mediated specifically by androgen receptors. And then these are not Brett's data. These are a recent replication of his study by my student David Zhang. David just defended his dissertation last week. This is looking at the number of songs in an hour between vehicle or cholesterol implanted animals. These are all castrated males, and DHT treated males, and the DHT levels treatment produce androgen levels that are comparable to intact males. And we also see some differences in the length of the song. And these mirror the kinds of differences we see with sex differences, where the number of songs is really sexually dimorphic. The length of the song is just slightly sexually dimorphic. And androgens didn't have any effect on frequency modulation or those other many of those other features. In fact, the one other effect that I don't show here is that androgens altered the starting frequency of the notes, and hopefully I'll get to that here in a moment. Okay. So in order to understand where androgens are acting, we wanted to map the vocal circuitry of the singing mice. And so to do that, we used a kind of cool tool. It's not a particularly new tool. It's not even very fashionable among neuroscientists these days, but it's a really powerful tool, I think. And this is a version of the chickenpox virus. It's called the pseudorabies virus that what it does is it gets taken up by nerve terminals and then transported back into the cell body where it replicates and then gets transported back again into the next cell body and continues doing that. Eventually, after about 4 days, it's lethal because it begins to infect and kill cells so much that the animal passes away. Um, but you can map out you can you can map out a pathway using this virus really quite quickly. And so when we do that, we see that that the motor. So what we did is we injected one virus into the larynx and another virus into the jaw, and then those are being coordinated with the notes. And what we see is that the laryngeal virus infects the motor neurons of the larynx. The jaw virus infects the motor neurons of the jaw. And then those start to come together here in the brainstem. They identify some central pattern generators. They go up then to the hypothalamus. And in fact, what we're seeing here on the left is a diagram of primate vocal motor pathways. And we see that the virus recapitulates those pathways nicely, going up all the way to the motor cortex and a variety of other areas. We look at androgen receptors and we see that if David triple labeled for pseudorabies virus and androgen receptors. And so what we're seeing in the middle is green is from the larynx, red is from the jaw, yellow is from both. And we're seeing regions of the extended amygdala, the hypothalamus, and these lower brainstem areas, all showing this evidence of being in this vocal motor path as we would expect. And then we see in the same areas androgen receptors, and then in fact, if we look at overlay those, we can see that they're triple labeled. So androgen receptors are present almost everywhere in the vocal circuit. And they're in the extended amygdala, the hypothalamus, the midbrain shown here. They're in motor neurons like the nucleus ambiguus. They're missing in the CPGs, the central pattern generators, which is interesting because those aspects of the songs are one of the places that there don't seem to be any sexual dimorphism. So in general, the distribution in motivational structures and motor structures corresponds to sex differences in behavior and to androgen effects on song. Okay, the next state-dependent thing I'm going to talk about is the influence of condition. And condition, we decided, wasn't very well measured by residual body mass like a lot of folks do. So we looked at a lot of different markers within plasma. Nutrients, hormones, a variety of things across wild animals. And we found that, did a PCA on those, and we found that the first component was sort of transient things that we might expect to be related to recent feeding or fasting. But then there are more stable markers, particularly adiponectin and leptin, which seem to be conveying something more stable about energy balance. And if we looked at those together with insulin and RBM, we could construct a composite score of condition. And it turned out that composite score was really predictive of song effort. And indeed, leptin alone was surprisingly predictive of song effort. So we did an experiment. Aaron Gilio just published this a month ago, or just this month, in Animal Behavior, showing that injecting leptin tended to add on to the effects of playback to increase the overall rate of song. So condition information seems to be influencing song rate in this meaningful way. So if we put all this back together again, we've mapped out all these circuits with the virus and found lots of cool things going on. I've suggested that there's this kind of pathway of things that are important for these display behaviors. And I just want to mention now that this particularly interesting structure here, these structures are being labeled as vocal control centers, sorry. And turn out to be the interface between kind of social information and energy balance information. So it's our hypothesis that this paraventricular nucleus within the motor path is actually a place for integrating resource holding potential, and will be a mechanism for that. I'm going to go ahead and stop now and just mention a couple brief things. Singing mice seem to adjust their vocal effort based on condition, predation, social context. They follow this general pattern I'm suggesting. And then we can use kind of systems neuroscience tools for investigating these decision mechanisms because these non-model rodents have such accessible brains. We're doing some interesting kind of brain-wide stuff that I'll be happy to tell you about, as well as some evolutionary genomics. But really what I should do is just close by mentioning, by showing some of the folks who have been involved in this work. Okay. Okay, my first question. Does sex ratio vary in natural populations? We have not observed sex ratio varying in natural populations, although I'm not certain. No, the difference in my sample size was not due to sex ratio. It's because we don't usually study the females' vocalizations because they vocalize so much less. Thank you. What's the social structure of singing mice? The social structure of singing mice is, so we think the mice can hear each other. If we do playbacks in the field, we can detect them. There's almost no noise during the day at these frequencies. And so we can detect them at about 20 meters. We haven't done playbacks of our field recordings to actually estimate the active space of the mice. But I think that that's actually plausible, which would be, that span would be far enough to sort of span more than one home range. So you'd be able to hear not just your neighbor, but maybe the animal past them. That's our best guess. The social structure overall is a general, like consistent with a general rodent social structure, which is promiscuous, with females being relatively solitary in their space use, and that being mediated by mutual avoidance from our experiments in the lab. Whereas males have lots of overlapping home ranges, but are still aggressive to one another. But they all come together at food sources like fallen logs. These guys are insectivorous. And then there's some suggestion that the xeromplenis, which are very low densities, might be socially monogamous, but we don't know that to be the case. Okay, next question from Fran. How much of a role do you think learning plays in the differences between allopatric and sympatric tiguina? I think it's all about learning. That's a great question. And the reason is that we don't really see strong genetic structure between those populations. They're like, they're pretty far from each other where we tested them, but they're not genetically distinctive at all. And so, yeah, what I really want to do, and we have a beautiful site in Costa Rica now where there's sympatry and syntopy, and I would love to manipulate their experience. We've just had a lot of trouble catching, Brett's situation, like that site isn't really accessible to us anymore. And so we've had a lot of trouble catching enough animals from syntopy to be able to really test whether it's learning or not. And the xeromplenis don't do as well in the lab as the tiguina do. So it's been hard to stage learning experiences as well. But I would certainly love to. And at some point, I definitely want to do that. Does age affect their singing behavior? We have looked at the ontogeny of singing behavior, and they don't seem to learn their song. It's a simple trill that has these kind of very rhythmic patterns, and they produce like we've done ontogenetic series of their singing behavior recordings, and like the very first songs we record look great, but they do kind of overproduce song just after puberty. Which would correspond to the time that they're dispersing. But beyond that, I think the singing behavior kind of plateaus. It doesn't, it declines with age slightly, but not dramatically.
Do we have any last questions? I'll give you a couple of seconds to decide. All right, then I think that it looks like we're good. Thank you so much for such a great talk. I really enjoyed learning about that. And I think a few of us have a meeting on the same call. So great. Everyone else, I'll see you next week.
Well, thank you all. I had a really lovely time sharing this with you.